Okay, today I want to talk about recent our results titled as Experimental Reconstruction of the Evolutionary Pathway Bridging Central Dogma Scenery by Bissing Linkford. And it has been almost 70 years since the central dogma was firstly stated by Francis Crick, but we still don't know the central dogma emerged, how the central dogma emerged in early life stage. But as you know, in the central dogma, the RNA polymerase transcribes the DNA into RNA, and then ribosome translates RNA into protein. Therefore, the origin of this machinery should be a key for understanding the origin of central dogma. But modern RNA polymerase and ribosome have very huge and complex structures with 400 kilodalton and 2 megadalton molecular masses, so very complex. And it's difficult to imagine such a huge structures emerge at very early life stage. So in that time, they must have been shorter and simpler structures, but we don't know what kind of molecules and how simple molecules they were. Looking at the structure of the RNA polymerase and ribosome, interestingly, both have small beta value structures as a important component. For example, the catalytic core of RNA polymerase is consisted of a six-stranded beta value structure called as DPBB. And similar but not homologous beta value protein fold rift is also conserved in ribosomal protein L3. And so far, previous studies propose that these beta value structure, DPB and rift, have evolutionary relationship, meaning that they have emerged from a common ancestral protein through divergence evolution. Furthermore, different beta value structures, SH3 fold and OB fold, are also conserved in many ribosomal proteins and translational factors. And similarly, it was suggested that Rift and OB and SH3, these pairs is resulted in the divergence evolution. So, however, probably because of very high sequence diversity between different beta variant folds, it has not been experimentally verified whether such drastic fold transition could occur in a feasible pathway. So, to uncover this problem, we experimentally reconstructed the evolutionary pathway between these seemingly unrelated beta variant folds. Firstly, I want to introduce the previous our research about the evolution of the DPB fold, this one, which is thought to be the core fold of RNA polymerase. And this DPB fold has an internal two-fold sheet symmetry. Left panel shows the topology of DPBB, and right is the tertiary structure. This DPBB fold is a six-stranded beta value structure consisted of two repeat beta, beta, alpha, beta unit. Each unit is shown as cyan and yellow here. So this DPB fold is proposed to have emerged by self-dimerization or by gene duplication of a shorter peptide with beta, beta, alpha, beta motif with 40 amino acid lengths. Take together this knowledge. Firstly, from a simple ancient peptide evolved to DPB structures through homodimerization and then evolve to repeat perfect symmetrical DPBB by gene duplication and fusion event. Then it further evolved to modern RNA polymerase through multiple structure complication steps. But in nature, there's no such molecular forces, so we try to reconstruct this evolutionary pathway and succeeded to create the perfect two-repeat DPBB and then dynamic half DPB like this. Additionally, by using this half-sized half DPBB, we attempted to create a more ancient DPB protein with simple sequence. And this dimeric half DPBB is consisted of only 30 amino acid types. So then we reduced the types of amino acid and asked how many amino acid types are needed to make this DBB structure. First, three hydrophobic amino acid, methionine, isoleucine, and leucines removed by replacing with valine. And then proline and serine were also removed by replacing alanine or lysine. And finally, tyrosine was also removed by replacement. So the final mutant consisted of only 7 amino acid types, and this mutant was named as delta mupis. And unfortunately, this delta mupis simplified sequence unfolded in solution, but forms crystal. And we verified this simplified sequence adapted into DBB structure in the crystal. So these results demonstrated the core fold of RNA polymerase evolved from dimeric peptide with about 40 amino acid lengths and 7 amino acid types. So very simple. Up to here, I talked about the early evolution of the DPBB fold. Then we wondered, can we convert this ancient simplified presumed ancient simplified DPB sequence to these defined beta barrier folds and reconstruct the evolutionary network? Firstly, we found a unique property of this simplified DPB that provide a hint for reconstruction of the evolutionary network. This simplified DPB delta mupis formed two type crystals at the defined conditions. One condition includes the malic acid, high concentration of malic acid, and another condition includes sulfate. And I said before slide, in the one condition, it adapted DBB structure like this, but I could not solve the second crystal structure by simple just molecular replacement using DPB model. From this result, I expected this delta mupis forms defined fold, not DPB structure. So then, what kind of structure is this? And previous studies suggested that there is an evolutionary relationship between DPBB and rift. So we guessed this unknown structures, maybe rift-like structure. This one. So this slide compares the DPBB and rift fold, and here I show the one of the presumed most ancient rift protein, PHS018. Anyway, both six-stranded beta-barrel structures consisting of two repeat beta-beta alpha-beta unit, but have different loop structures. In the case of DPBB, the first loop forms and colored as pink formed the side loop. But in the case of rift, this first loop forms a simple beta turn. So, and this defined loop connection resulted in the changing the second beta strand direction oppositely. This one. And yeah, comparing the sequence of N and C terminal half of rift and simplified sequence, and their sequence similarities are near 50%, very similar. So we try to create the intermediate chimera of DPB and rift protein. The 5 residues specifically conserved in the rift protein was introduced to the simplified DPB sequence, and the chimera protein was named PH1 here. And we confirmed that this PH1 formed stable dimer and solved its crystal structure like this. And unexpectedly, this PH1 adapted into a new novel beta barrier structure, neither DPBB nor rift. And the most defined point is the configuration of the first loop. It was rolled up, unlike the DPBB and rift. And because this loop configuration resembles the letter of zeta in the topological scheme here, zeta. So we named this new beta barrel structure as double zeta beta barrel DZBB. It's complicated. But anyway, this discovered new structures allow me to solve the second type crystal structure of the simplified sequence consisted of 7 amino types. It also adapted DZBB structure. So this simplified sequence can form two defined beta variable structures, not only DBBs, but also DZBB structure, like metamorphic protein. And 5 additional mutations from modern lift protein stabilize only DZBB structure like this. If this DZBB structure, new beta variable structure, is an evolutionary intermediate between DPBB and Rift, it should be easy to convert to Rift fold. This allow. So I further engineered this DZBB protein to convert to Rift fold. The difference point between these beta variable folds is only the first loop configuration, this one and this one. So I engineered this sequence. Today I skipped the detail of the design. Anyway, this loop, zeta loop sequence was modified to form beta turn like this. Structure analysis of this mutant revealed that it actually formed the homodynamic rift fold, and engineered loop made a beta turn expectedly. So this DZBB could be converted to rift structure by simple mutation. Up to here, we showed that it's easy to fold conversion from DPBB to a new beta variable structure DZBB and then rift. This means that this DPBB and Rift may have evolved through DZBB structures as a evolutionary intermediate missing link in the fold evolution. Next, how about OB fold other beta variable structures? Very interestingly, we found somewhat similarity between DZBB and OB fold structures. In this slide, I compared both these beta variable folds. And when superimposing the DZBB, in particular its monomer with OB domain from ribosomal protein S1, the 3 beta variable strands, beta 1 and beta 2 and beta 3, were overlapped well. Additionally, comparing the topologies, the beta 2 of OB is absent in the DZBB structures, but the other regions are almost identical, and the sequence comparison also showed relatively high similarity. The homology is over 30%. So we therefore created a chimera of DZBB and OB protein. Here I use the sequence of OB domain of ribosomal protein L2. I also skipped the design details, but basically we incorporated the sequence around the beta 2 into the DZBB protein. And the ratio of DZBB and OB is 4:6. And the two designed chimeras folded in solution, and their crystal structure could be determined like this. Yes, the chimera adapted into the OB fold expectedly. And we also created chimera with higher DZB content ratio, 6:4, and they also adapted to OB fold like this. Yeah, we solved its crystal structure. And so therefore, the chimera between DZBB and OB, like an evolutionary intermediate, could be reconstructed like this. In other words, this DZBB fold was a missing link in the fold evolution, linking not only DPB and Rift, but also OB fold. Finally, we tried to connect SH3 fold to this beta variable evolutionary network. And it was previously suggested, maybe there is Lauren Williams groups. Oh, yeah. Thanks. Okay, and yeah, his group previously suggested that the interconversion from OB fold to SH3 fold occur by a simple circular permutation. So maybe he will talk about this kind of structure transition something. But anyway, following this, his hypothesis, we permutated this DZBB-OB chimera. The C-terminal sequence was firstly cut here, and then connected to the N-terminal end with two residues, which forms a beta term, this line, and yes, and we confirmed that the bottom mutant MKL2SH3, this mutant fold well in solution and solved its crystal structure like this. Yes, the mutant adapted SH3 fold expectedly. So therefore, we succeeded in the fold transition from OB to SH3 fold by simple permutations. Yeah, taken together, these results, DPBB, Rift, OB, and SH3 folds share the same origin and likely emerge by the fold interconversion through DZBB fold, like a missing link fold. And this evolutionary trajectory needs only a few mutations. And firstly, the simplified sequence consisted of only 7 amino acids types, formed two structures, DPBB and DZBB, and the additional 5 mutations stabilized only the DZBB structure like this, and the transition from DZBB to Rift can be occurred by only one single sequence deletion and a point mutation, only one point mutation. And this view could be also converted to OB fold by sequence insertions with 7 amino acid lengths and only 4 point mutations. And this OB protein can be converted to SH3 fold by simple circular permutation. So this indicates this easy to convert the fold transition implies that these divergent evolution have been completed in a very short time. So this is the last slide. Yes. Yeah, and our results indicated that the demonstrated the simplified sequence consisted of only 7 amino types can form the protein structures, and it adapted two structures, DPBB, not only new beta barrel structures. And we also reconstructed the evolutionary trajectory between distinct beta barrels, DPBB, Rift, OB, and SH3 folds through new fold, DZBB, a missing link fold. And these results also indicate that RNA polymerase in the transcription and several ribosomal proteins, so translational proteins, have arisen from a common ancestral peptide in early life. So, and it also indicated that the divergence evolution of important building blocks in the central dogma have occurred in a very short time. That's all. Yeah, thank you very much.